Well, thank you, everybody, and welcome to Strait Science. Strait Science is an evening seminar series put on by UAF, University of Alaska Fairbanks Northwest Campus in Nome, and UAF Alaska Sea Grant, also in Nome, and tonight you're in the home office. So tonight it's pretty interesting. Talk about a Strait Science. We're actually going to talk about these nice people are going to cover where we are. So, and give us a little history, a look into the past. They're doing some research on SIKULIAQ this summer, which they're also going to mention. And we're excited because we'll see the ship here in Nome, and they have promised to come back and give us the more information as it unfolds. So we're really excited and glad they are here. We have 3 speakers tonight. We're going to lead off with Sarah Fowell. She's the lead of their project. She's in Fairbanks, so she's on our time zone. She is a paleontologist at UAF Department of Geosciences. The other 2 speakers will talk more about themselves, but I'll just mention it here. The second speaker will be David Scholl. He is in Ohio, so it is currently 10:15 for him, or sorry, 10:30 for him. And we appreciate you dealing with that time zone and taking your time to talk to us. He is emeritus. He was the senior scientist at the US Geological Survey, and he's marine geophysicist. Would we say that, David?
Yep.
Okay. And then bringing it all home will be Beth Caissie. She works with the Pacific Ocean Paleoclimate Group at USGS, US Geological Survey. She's in Menlo Park, California, so she's got an hour difference too. We are all over the United States at this point with you guys. So what was the Bering land bridge like? Something to think about when nowadays we're all focused on all the changes in the Bering Strait, but I think the 3 here are going to put it all in a little bit of a different perspective. So I appreciate that, and we're very interested. I'm going to leave it at that, other than to say I don't see any callers on, but no, as people at Strait Science do, if we get a caller, they will have priority in questions, and because it's very hard to be a caller on a Zoom call, as we know. With that, Sarah, welcome to Nome, and take it away. Thank you so much for coming.
Thank you, Gay. I'm excited to be here. As Gay said, I am Sarah Fowell. I'm coming to you from the University of Alaska Fairbanks, and I want to acknowledge that the Fairbanks Trothyadak campus is located on the ancestral lands of the Dina people of the Lower Tanana River. So let's talk Bering Land Bridge. Over the last 2 million years or so, glaciers have expanded over Earth's northern hemisphere more than 20 times. And during these ice ages, a lot of Earth's water was tied up in ice sheets, causing sea level to fall. Lower sea level led to emergence of the Bering Land Bridge above sea level. And that Bering Land Bridge served as a migration corridor for plants and animals passing between Eurasia and North America, or North America to Eurasia. This map shows the extent of the land bridge about 18,000 years ago during the height of the last ice age. This is a time known as the last glacial maximum. And here, regions that are currently below sea level, but 18,000 years ago were emergent exposed land, are shown in yellow. And you can see the land bridge in this region. Bridge always seems like a funny term for it because you think of something skinny connecting 2 areas, but this bridge was more than 1,000 kilometers from north to south. It was quite wide along the southern coastal region and the northern coastal region. And plants and animals that were living on the land bridge didn't know they were going anywhere. They weren't trying to get somewhere. This was their home. Now, for a long time, we've often assumed that the vegetation on the land bridge was much the same as what we reconstruct for the Ice Age vegetation of Alaska or Siberia. And that would be something like this. A vast steppe, an arid grassland, somewhat similar to the subarctic steppes of Mongolia today. And that's what I'm showing you is a picture from Mongolia, but we think that the Bering Land Bridge may have looked a lot like this. Certainly Alaska and Siberia had stretches of vegetation that was much like this. You'll notice the lack of trees and other woody plants on this landscape. Some differences: the Bering land bridge would have been populated by horses, woolly mammoths, bison, and musk oxen instead of camels. But I work a lot in Mongolia, and I thought I'd talk a little bit about how we propose to reconstruct what the vegetation of the Bering land bridge was like in the past, and then I'll tell you why it's an interesting question. To use pollen and spores to reconstruct past climate and vegetation, I typically collect long sediment cores from lakes. Pollen and spores produced by plants growing around the lake were deposited with the sediment at the time of deposition, and the organic matter in the sediment can be dated because it contains carbon-14, so we can radiometrically date it. So I know how old the different sediment layers are. I can take sediment samples, through a chemical process, remove fossil pollen and spores, and make a slide of assemblages from different times in the past, and then look at how the proportions of different spores and pollen grains changed over time. In the steppes of Mongolia, sometimes those changes are minimal. There are a few trees or shrubs, as I mentioned before, and these three groups tend to dominate. Here we're looking at sage, or the genus is Artemisia pollen. It's really thick walled. It's got sort of three linear slots or apertures where the pollen grain may germinate. This is goosefoot, or the Chenopodiaceae family. Their pollen grains are really fun because they've got little pores all over them, so they actually look like tiny, tiny little wiffle balls. And then down here is grass pollen. And let's just hide these tools. Down here's grass pollen. This is all pollen of the grass family, looks pretty much the same, which is a little disappointing because it means it's really hard to tell different species of grass apart based on their pollen grains. And different species do have different environmental preferences and requirements, but all pollen of the grass family pretty much has one nice round sort of spherical objects with one single pore. So these are very common in records from Mongolia, also very common in Alaska and Siberia during the ice ages. And what this is saying is basically sage and goosefoot pollen are more common in dry climates. So plants are more common in dry climates. Grasses become more abundant when conditions are a bit wetter, when there's a bit more precipitation. And that means if I'm looking at a slide and I count percentages of different pollen types, and I add the percentage of sage and goosefoot pollen at any time in the past and divide by the percentage of grass pollen, I get a number which increases when conditions in the past were drier and decreases when conditions were wetter. So even from these three taxa, by looking at their relative proportions, I can pull out a climate signal, which is kind of cool. Okay, so that's what I do for my research. Come on. There we go. So back to the land bridge, a puzzling thing about the Bering land bridge is that even though animals could walk from Siberia to Alaska or vice versa, some, such as horses and woolly mammoths, did that. They crossed the land bridge, but others, such as musk oxen, did not. And we're not sure why. The two main possibilities are, firstly, bad timing. Maybe the animals arrived at the margin of the land bridge at the wrong place or at the wrong time. They weren't wound up in a place where you couldn't cross, where the water was already rising. So they could have just had bad timing. The second possibility is that some sort of barrier, maybe an ecological barrier, prevented certain animals from crossing, but not others. It's something known biogeographically as a filter bridge. Some animals got through, some did not. And the barrier idea is getting some support. It's been suggested that this barrier might have been a difference in vegetation between higher elevation regions in Siberia and Alaska and the lowlands of the Bering land bridge, which here are shown in white. Specifically in 2001, Dale Guthrie, also of the University of Alaska Fairbanks, called the Bering land bridge the mesic, or that's a word for relatively humid buckle in the arid steppe belt. In other words, he proposed that the lowlands between Alaska and Siberia were wetter and contained more tundra adapted species. And this limited the distribution of arid adapted animals that fed on the arid grasslands. They weren't finding a lot of that on the lowlands of the Bering Land Bridge, and so they didn't cross. And those animals are shown here in these green arrows. The rhino, the camel, the kayang, the bear, the muskox, and the badger are animals that got right up to the edges of the Bering Land Bridge, but for whatever reason did not cross to the other side, which is a continuing source of disappointment to me, because I would have really liked there to have been woolly rhinos in North America, and specifically in Alaska, but we did not get them. So again, here the Bering Land Bridge, the lowlands are shown in white. This is the area that's under sea level today. These blue blobs and polygons are actually sedimentary basins on the Bering Sea floor, which may have been, or probably would have been, sort of low areas, topographically low areas, possibly housing lakes or bogs during times when the land bridge was above sea level. And I'm going to talk more about those in just a couple minutes. This green polygon sort of outlines the edges of Guthrie's proposed more humid area where tundra vegetation might have been more common. Okay, here's a little summary of what paleontologists know and how they see our knowledge of the Bering Land Bridge. This was put together by Elias and Crocker in 2008, and it summarizes basically mounting evidence for Guthrie's idea, mounting evidence for a belt of relatively humid tundra vegetation covering the lowlands of the Bering Land Bridge, which here are shown in this light green color. Siberia and Alaska are a little bit darker green. The dots represent locations, and these locations are mostly lakes that have been cored. And from those cores, we've obtained fossil pollen grains from plants that were present on the landscape during the last ice age. So all of this pollen evidence, and in some cases, we've also got fossil insects, mostly beetles, from these sites. All of this evidence comes from mostly lake cores. So the yellow dots on the map, these yellow dots, are places where the vegetation appears to have been relatively dry steppe, with few or no trees or shrubs. The red dots, we've got some here, over here in Siberia. The red dots are places where the climate and the vegetation appear to have required more moisture. The plants required more water, and the insects did as well. Now, generally speaking, there's some evidence from diagrams like this that things got a little more humid, a little bit wetter as you approach the margins of the Bering Land Bridge, but the data doesn't completely agree. For example, fossil pollen from high spots on the land bridge from islands today in the Bering Sea, such as St. Lawrence Island and St. Paul, record relatively dry steppe vegetation. And this is particularly surprising for St. Paul Island because we're down here close to the coast, close to the southern coast of the Bering Land Bridge, which you might have expected to have a more maritime climate. And from, maritime climate areas, I mean, let's look at Alaska today. When Guthrie proposed the idea that things were a little bit more humid on the lowlands of the Bering Land Bridge, when he suggested that that area was the mesic buckle in the arid steppe belt, he noted that tundra, here's sort of just general distribution of tundra today. Of course, there's different types of tundra, which the second figure identifies. But he noted that tundra is present in coastal regions today due to frequent cloud cover. It's certainly possible that cloud cover shrouded low-lying coastal regions of the Bering Land Bridge as well. And right now, if we're looking at islands, we're only looking at slightly higher elevation spots. So what we really need in order to answer questions about the vegetation of the Bering Land Bridge is samples from lakes or bogs on that land bridge when it was above sea level. And there are two problems involved in obtaining these samples. The first is that those lakes have subsequently been covered by the ocean, covered by the Bering Sea as glaciers melted and sea level rose. It's hard to, and okay, so they're underwater. Makes sampling a little bit difficult. And it's also hard to know where lakes would have been on the Bering Land Bridge, but we have some ideas. And in 2019, Jeffrey Bond of the Yukon Geological Survey published this paleo drainage map of the Bering Land Bridge. And what it actually is designed to show you is likely locations of rivers, lakes, and bogs. So standing water bodies are what we're particularly interested. These would be places where sediment, spores, and pollen grains would have been accumulating sort of in an annual fashion. So a great record of what plants were living around those lakes during the last ice age. Now, if Bond is right, and if we could collect cores from these lakes, sediment that was deposited in those lakes when the land bridge was above sea level, we would have a much better idea of what the vegetation of the land bridge was like, how it changed from north to south, because this is a really big area. It's unlikely that it was all the same on the land bridge, and maybe how it differed from the relatively high areas of Siberia and Alaska to the east and west, and whether woody plants were indeed present and in what kinds of proportions. So I mentioned that this area is currently flooded, and any sediment deposited in lakes and bogs on the land bridge is now under marine sediment, but that doesn't mean that lake sediment is inaccessible. This map was compiled by one of my collaborators, Jenna Hill, and it shows the thickness of the interglacial or the Holocene marine sediment package at various locations in the Bering Sea. The red and orange areas are thicker areas of sediment, more than 8 meters thick. The sort of blue and greenish areas are places where the marine sediment is 4 meters thick or less. Now, if we were out on a ship in the Bering Sea using a gravity core, which is just a big tube, a long tube that falls down toward the sea floor with weights on top of it, so it's falling under its own weight and forces of gravity, those tubes can collect a sediment core that might be 10 meters long or more, maybe even up to 18 meters long enough to see what's under those marine layers. And what's under those marine layers is stuff that was sediment that was deposited on the Bering Land Bridge. So here's the plan. As I mentioned earlier, about 5 sedimentary basins, large sedimentary basins, have been identified on the floor of the Bering Sea. These would have been low spots when the land bridge was above sea level, and it stands to reason that they could have been sites that housed freshwater lakes or bogs. Low spots is where water tends to accumulate. And interestingly, several of these basins, especially these northern ones, the Hall, the St. Matthew, and the Norton Basin, are places where Bond actually placed lakes on his paleo drainage map. So it also looks like these are likely locations for lakes. Therefore, in order to directly sample sediment deposited in those basins during glacial stages and in lakes on the land bridge during glacial stages, my colleagues and I from University of Alaska Fairbanks and the USGS have received funding from the National Science Foundation to collect a suite of cores from across the Bering Sea shelf. Basically across the southern half of the land bridge. And we're going to collect these cores for the purpose of reconstructing the ancient ecosystems of that Bering Land Bridge. We're currently scheduled to sail on the research vessel SIKULIAQ during August. And this sort of orange line on this map, that is the general path that our ship is going to take. So we're going to be collecting cores across the Norton Basin, across this region where we can find the St. Matthew Hall basins, across the Navarin Basin, across the St. George Basin down here south toward close to the edge of the shelf and close to the southern coast of the land bridge, and here on the North Aleutian Basin. So fossil pollen in those cores can help us to answer these long-standing questions. What plants were present on the Bering Land Bridge during the last ice age? How did changes in latitude, elevation, and distance from the coast affect those plant communities? In other words, how did the vegetation change at any one time across the land bridge? And what resources were available to animals that lived on the land bridge? Is this the barrier, just the food that was available? Is that the barrier that prevented some animals from crossing? In addition to looking at the fossil pollen in the cores, we are collaborating with colleagues who can extract fragments of ancient DNA from the sediment in order to find evidence of what animals were living near the lakes or the bogs. We won't find actual animal fossils. We're not, even if we get the DNA, we won't get it from remains of the animals. The cores are going to be too small in diameter to sample anything except really tiny fossils such as pollen grains. But we can find bits of DNA if the animal was urinating or defecating near those lakes. And some of that ran into the lake, we might be able to pick up their DNA, and then we would be able to say that those animals were present. So we should be able to reconstruct both plants and animals present at different locations across the Bering land bridge during the last ice age. And now I'm going to turn it over to Dave Scholl to tell us how we'll finalize site selection and actually identify lake deposits that are beneath the marine sediment layers on the Bering Sea shelf. So,
All right. Thank you, Sarah. We did have a question in the chat, so I don't know if you want to wait till the very end or you guys want to answer the questions that come during your segment.
and this land bridge was present multiple times over the past 2 million years. It would have been present for, it depends on the exact ice age, but sometimes 10,000 years or more at a time during a given ice age. It would flood during an interglacial stage when things warmed up. As ice sheets expanded, the land bridge would slowly become exposed. And it was present at its maximum extent, which I was showing you, probably for several thousand years.
And that's the answer to the question, how long did the land bridge exist? Yeah.
And
Awesome.
if I can just add to that, that a paper
Yes.
just came out that had it, the land bridge exposed for about 20,000 years, most recently from like 11 to 34,000 years ago.
Wow. Okay. Well, thank you. And I think that's it, unless there's any other questions. I see none. Take it away, Dave. Thank you.
Yeah, I'm gonna try here. I'm gonna see what kind of magic happens here, but I have to get rid of this because I can't see the share screen. Yeah, I'm not sure this is gonna work for me.
If it
And,
helps, just look for that,
Yeah.
the word
Yeah, you might
view
want
in
to go up to
silver.
the view thing again.
Again.
Sometimes after someone else shares, things get reset. So
So-
exit full screen and it should show up.
Yeah, we had this problem before, and I got rid of the, you know,
Yeah, I think if you go up to that little 9 dots in the upper right-hand corner where it says view, can you exit full screen?
yeah, I'm gonna have to do that. Let's see if I can catch up with you guys. Okay, I'm gonna click on it again. Okay.
No
and,
worries. No worries.
Okay. Now, can you see that? I doubt it.
No, you need to be seeing the toolbar with the green button at the bottom or the side, wherever you may have.
Yeah, that's what I'm not finding. I'm going, oh dear, where the heck did that thing go? Yeah, let me look around here a little bit,
You,
lift things up. If not, we'll just not worry about it.
well, while Dave's doing that, I want to say this has been already, I've learned heaps. I really appreciate that. I didn't really recognize the importance of pollen. And I've got some questions for you when we're done, but that is fascinating that you can get the ancient DNA out of a core, however many thousands of years old from the, amazing. So
Yeah,
very-
you can
Yeah.
get DNA, little snippets that are particular to certain groups of plants or animals. It's actually really great to use that in conjunction with the pollen and spores, because sometimes you have very tiny amounts and you have to ask questions like, did these blow in from very far away or were they local? And if you find the DNA of that plant, that doesn't travel well. And then you can say that it's local.
So you got another question in the chat saying, how can you confirm that you are actually pulling cores from former lakes?
Yes, and part of that is the sediment itself. And some of the isotopic signatures in this sediment are typical of lakes versus marine systems. But when we find nice packages of layered sediment, we know we're finding a low spot where sediment was accumulating on a fairly regular basis. Those tend to be underwater, so they're likely to be lakes. If they're on the land bridge when it was high and dry, we should be looking for fine-grained sediment like silt. We should be looking for lots of organic matter. But our isotopic signature will also tell us whether it was a freshwater or marine system. Right on, Dave.
Dave, you're
Yeah.
on. You're on the
Yeah,
screen. You made
yeah.
it. Now you
I
just
found-
need to.
It was hiding. I just. had to
I've
run
been
it
there.
down.
I've been there. It always does it when it's your turn. That's the thing, isn't it?
Oh, yeah. Well, hello, everybody. I'm in Ohio, so I'm past my bedtime a little bit at any rate. But what I wanted to talk about a little bit was how we can image beneath the seafloor, the Bering Shelf, to see areas that we would like to take cores, wherever there might be lake deposits and things of that sort. How we can map the seafloor and decide where the vessel should take its samples. Now, I have a movie here I was going to try to show, but it might not work, about the flooding. But this particular image here, at any rate, and by the way, can you see my cursor, everybody? Can you see that guy?
We can. We
Yes.
can. Yep.
Okay, that's good to hear. At any rate, this is sort of a long view from the North Pacific, where the water's quite deep out here, 20,000 feet or so. Here are the Aleutian Islands, and this is the so-called Aleutian Basin. It's deep water, Bering Sea, 13,000 feet or so to the seafloor. But up here in the shelf, the land bridge, it only gets down to about 500 feet at all. That's the Bering Shelf, and that has been called the Bering Land Bridge. So when a lot of ice was on the continent, a lot of glaciers are on the continent, a lot of water was extracted from the oceans to do that, and the water withdrew from this entire shelf area, and it was dry land, and that became the Bering Land Bridge. And it was a pathway for animals of all sorts of plants, I guess, too, to some extent, but also people. And a little bit of information about that before I go a little further. But this is I had a little fun putting this cartoon together, and I've got mammoths out here on the Bering Land Bridge. I've got steppe horses. I've got steppe bisons. I've got stellar sea cows. And this is just a little picture of a tusk, a mammoth tusk found on St. Paul Island. So they certainly got out there. So these are some of the critters that were running around out through here. And if you're wondering about whether people crossed here, some new evidence coming forward at any rate. And this is in New Mexico at White Sands. They have found footprints of people here. You can see them. They were not wearing shoes as such because their toes are nice and straight. They really look cool. And these are being dated in at around 21,000 years ago. So this is during the last ice age, when sea level was very low. People were making their way into the Americas. This is provisional dating, so we'll see where this goes. But these footprints are in association with mammoth footprints, camel footprints, giant sloths, all those sorts of guys who lived in the last ice age. Now, sea level back here 120,000 years ago was a little bit higher than it is now. But as the ice age came on, the last one, it went down and down, little bumpy, 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 bumpy. And somewhere around 18 to 20,000 years ago, it got about as low as it could be. The Bering Land Bridge was completely exposed, basically all the way out and beyond the Pribilof Islands. And then the melting started. And the melting started, and sea level starts to rise, and it goes up here. And this, it represents the running of water, the flooding of the land bridge until what we see now, of course, off in the Bering Sea. So it went all the way up here, it sort of leveled off a little bit. But this is the, hello here, Bering Land Bridge. Go back where you belong. Okay. Now, I'm not sure this movie, Sarah's going to try to run it. And if you're going to give it a shot, Sarah, give it a shot. Shall I click out?
And yeah, just stop the share. And I think
Yeah.
that's gonna, I think that's
Okay.
good.
Okay.
Yes.
And the blue is the water, of course. And here we go. This is put together by InStar, University of Colorado. And you can see the blue, and we're at 15,000 years ago. And it's coming over here on sort of in the Gulf of Anadyr side. Water will come up through the Bering Strait. And around 10,000 years ago, basically, we're free flooded. Now, this diagram was made based upon the depth to the seafloor right now. But as Sarah was pointing out, a lot of sediment and many meters of sediment have accumulated on the Bering Shelf. So this diagram doesn't necessarily represent an accurate view, but a good one at any rate about how the Bering Strait was flooded about 10,000 years ago. And of course, Nome is right over there. So that's sort of how it looked. And whoever was out there at any rate, either you started to walk across right about now on ice in the wintertime, or you got a boat. But the animals were pretty much in a bind getting across here about 10,000 years ago, one way or the other. Okay, Sarah, thanks much. I'll go back and screen share here. Okay, if I can get this done. Okay, then I will go to my next slide.
We
The
can see it.
devices that
Perfect.
we have on, can you see it? Are we okay?
We're good.
Okay. We're looking at it with a chirp sonar technique that the SIKULIAQ has. And most of us are sort of interested in sonar. We've heard all kinds of, Hollywood movies of a submarine and a destroyer passing over it, and it goes ping, 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 ping. Okay, that's a simple little ping. But when you go into a chirp mode, you go from a low frequency to a higher frequency, and it's much longer. But it's not a ping, but it's a pulse. So it starts out like going ping, ping, and you can collapse that into some really nice, cool techniques at any rate to really get details. And what this shows here is a fish finder. This is a little old-fashioned ping, and everything's kind of fuzzy. But when you go on the chirp, at any rate, you can see that. Oops, didn't do that very well, did I? You can break the fish down, and you can see them. And this is what the device looks like. Stop that. This is what the device looks like in the bottom of the SIKULIAQ. That's where the sound is generated. And this is the little magic box. There is a different technique, and we'll be using this one primarily. This is a Kronosberger. It's what they call a Topaz PS18. It's a little stronger. You can get a little deeper penetration. And this cartoon shows you that you have these various beams that go out. So you get a pretty good look at things. And this is what the sub-bottom looked like. Here's the seafloor, and these are various layers underneath the bottom. And our job is to find out what these layers are and looking for lake deposits, bogs, and things of that sort. And this is just the onboard electronic components. Now, what does a good chirp record look like? It looks like this. Here's the seafloor here, and this is off of Cape Navran, off of Russia at any rate. Water depth at this point is 100 meters, 100 feet rather. And the sort of pale gray here is the marine layer. That's the material that was deposited after sea level rose across this area. But beneath that, down into here, these banded things, that was part of the land bridge, whatever those deposits are. And as Sarah was pointing out, that's our job to find out. And there are other things that are kind of diffuse. They might be sand dunes. They could be most anything. There's a lot of learning that goes on. We'll be stopping the SIKULIAQ in places and taking cores down through here. This is out in fairly deep water, and the sediment is kind of thick. We might not, well, we'll see what we can do out there. But here's another place where the water is only about 30 feet deep. And this is off Nome itself. And the sand layer is rather thin, and we'll be able to go back down through that. And we should be able to punch right into these land bridge sediments, whatever they are, whatever they turn out to be. A lot of currents in this area, so you can see that the seafloor is eroded here at a rate. So that's basically it. We'll be doing basically sonar, the sort of stuff that you would normally use to find fish, but with a little stronger, little lower frequency, and we then can penetrate the seafloor, see the layers, take the samples of them, and learn an awful lot about what's down there. And we'll get smarter as we go along and learning to interpret the records, where we should be, taking our cores. Okay, that's really all I have to say. And I'm glad that Sarah was able to get the movie going. That worked out okay. Okay, I'll click off now.
All right. Well, thank you so much. I don't know if that is an animation that's available to the public, but I would put the link if you can to in the chat box. Just copy that link, or maybe Beth can do that because she was running it when she's done with her.
It's available. It's you can get it. It's no problem at all.
Yeah, it was wonderful. So there is a comment from Rick Toman that from the animation, the final breach that connected the Bering Sea to the Chukchi Sea occurred between St. Lawrence Island and Chukotka. So that's pretty interesting for us. If you look right here.
Yeah, and we're not able to go over there.
We're familiar with that problem, but yeah, that's very interesting. I was up looking at where Diomedes are, so that was very interesting how that kind of opened up, what that must have looked like.
Yeah.
So, all right. Any questions for David at this point? Burning questions? I think Beth is set up and good to go.
Okay, you can see my screen, I take it. So just building off of what Dave was talking about, this is some examples of some of the cores that we might actually end up taking. I don't know if you can see my cursor here, but
we can.
Okay, there's a couple of different types of cores that we're going to be taking. So this one on the right is a gravity core, and so it just sort of gently goes into the sediments. And on the left is a piston core, which has a hydraulic trigger that pushes it into the sediment with a lot of force. And we also have a vibracore that will vibrate as it goes in, and that will be a good one for taking sandy sediments like we think we'll probably find off of Nome. And so we can take these cores onto the ship, we can cut them up and look at them. And what we'll eventually end up doing is taking the cores and slicing them lengthwise. And then, let's see, I have to figure out how to advance here. Slicing them lengthwise and then just opening them up essentially like a book and reading those sediments. And so in this core, well, actually, let me back up for a second. If you think about the sediments in the ocean, anything that's out there in the ocean is going to eventually die, fall to the seafloor, and start to accumulate. Large things, small things, things will break down, rock, sand, stuff that gets blown in by the wind. And so, a little bit will accumulate, and then a little bit more, and a little bit more. And so what you end up with is the sediments on the top are really recent, and then as you go down, they get older and older. And so if you look at this core, it has a lot of different colors and structures. There's some volcanic eruptions in this core. And so you can see a lot of environmental change that's happened just by looking at the core with the naked eye. But what we're actually going to then do is use the microscope, and Sarah can look at things like pollen and how that pollen changes in response to changing environmental conditions. And we can also look at really small organisms called microfossils and how they changed with changing environmental conditions. And so for me, I'm really interested in the sediments on the top. So Sarah's interested in the land bridge, and I'm interested in what accumulated since the Bering land bridge was flooded, the marine sediments, because I'm interested in sea ice. And anybody in Nome, you're no stranger to changing sea ice. I got this picture a couple of days ago and I really love it. Here's Nome over here on the left. And there's a couple different types of sea ice that you can see here, and I'm going to come back to that in a little bit. But when you think about sea ice from the context of the entire Arctic, sea ice has been declining since we started watching it with satellites in 1979. And so this graph, the red line, is showing you the extent, the area of sea ice in the Arctic Ocean and how that has changed. These are only in September. So this is the summer sea ice, the sea ice minimum. About 2000, sea ice really started to decline a lot. And in 2007, it took a really steep dive. And we expect that eventually we'll have ice-free summers in the Arctic, which is, I think, kind of a terrifying thought, and it's going to be a huge change. And so the thing that I think about all the time in my work as a geologist is, what did sea ice look like before 1979? And, you know, what did sea ice look like as we came from that last glacial into the Holocene today? So as it warmed from out of that last glacial, how did sea ice change? So this poses a question, like if we've got sea ice, which, you know, anybody in Nome knows, it advances in the wintertime, and there's lots of sea ice in the Bering Sea, and then it pulls back and melts in the summertime, and there's no sea ice in the Bering Sea. So what does it leave behind so that we can tell whether there was sea ice there in the past? So if we think about the sea ice ecosystem, you might initially think about polar bears or seals or maybe walruses. But what I'm looking at is the ice algae. And this ice algae attaches to the underside of the ice. Sometimes it actually can live within the ice. There's zooplankton that lives within the ice too and eats the ice algae, and fish are going to eat the zooplankton, or they're going to eat the ice algae. There's actually so much of this algae that it falls to the seafloor, and then a whole lot of things down here also eat it, like crabs or sea urchins, maybe sea stars. And so it supports this entire sea ice ecosystem, this algae. If you were to take a core from sea ice, just go out there and, you know, like use an ice auger and take a core, the bottom of it might be brown in the springtime. And it might look like this. It might look like this if you went to swim underneath the ice with lots of this filamentous algae hanging down. And so these are actually diatoms. This is the most common algae in the Bering Sea. And some of them are living in these little channels that form in the sea ice, that as the sea ice is freezing, it gets rid of all the salt in the salty water, and it makes these little channels where the salt goes out, and then diatoms and other algae and zooplankton can live in those little channels. So if you were to look at this, melt this, and look at it under the microscope, it might look like this. These are all living diatoms. They're green because they photosynthesize, so they need light in order to live. And they are single-celled algae. So each one of these little hamburgers here is an individual organism. They connect to each other by threads, and they form these colonies. Or this particular rectangular one connects to each other. They just stick to each other's cell walls to make a colony. But the thing that is the best about them, if you're a geologist, is that they make their cell walls out of glass or silica. And so they are actually preserved in the sediments for hundreds of millions of years. And as you can tell, they come in lots of different types. And so the different species change depending on your environment. So for example, if you were to go out in the Bering Sea in an icebreaker in March, right after the light makes it through the sea ice so that you're photosynthesizing, you might see some diatoms on the underside of the ice, and they might be this species, or maybe this one. You might have some colonies of this Nichia. But then if you were to go out a little later, a couple months later, the ice is starting to melt, it's starting to break up. Instead of having a solid coating of ice, maybe you have some water between the ice, between the flows, and you might have a whole different set of diatoms that are living. Instead of the ones that are attaching to the underside, you might have ones that are living right on the edges of the sea ice or in the water around it. And so now we might have these different species. And so we can think about this on a short time scale. So what happens from, you know, March to May in a year? We can think about it in space. And so if we come back to this picture with Nome and the different sea ice, maybe the species that are living in the pack ice are going to be different from the species that are living where the ice starts to break up a little bit, or the species that are living in ice that has water, that's living in the water around that ice. So short time scales, we can think about it on space, and then we can think about it on long time scales. So if we look down in those sediments and we see lots of, say, this guy along with this one, we might say, oh, there wasn't that much ice at that time. Maybe it was only present for a couple months per year. Whereas if we see a part of the sediment core that has lots of these species, we might think, oh, there was a lot of ice. Maybe it was around for 10 months per year or something like that. And so when we get that information from a bunch of different cores, we can start to compile what happened, what did sea ice look like in the Bering Sea. And so this map, this is Alaska and Russia over here. And the GIF starts at 22,000 years ago during the last glacial. And you can see how this is the sea ice, is the white blob, is changing as sea level rises, as climate warms, as we come out of that glacial. And what's interesting about this is each one of these dots here is a sediment core, and it's a sediment core that was taken from more than 3,000 feet of water. So sea ice does not form in 3,000 feet of water today. It's forming only on the shallow water. And so what's exciting about this cruise is that nobody's ever taken sediment cores from the shelf before to look at changing climate. And so that's what we're going to be able to do. We're also going to be able to ask questions like, does sea ice have a control on aridity on land? And so if you think about how arid or mesic or moist it is on the continent, there's a couple of ways that you can change that in Beringia. And so one way is you can reduce evaporation. And so if you have sea ice out here, this is not very much sea ice, or you can add a lot of sea ice. If you have a lot of sea ice, you're covering a lot of the ocean, and so there's less ocean available to evaporate. Or if you have sea ice lasting for a long period of time, then you have fewer months per year when evaporation can happen. The other way you can change aridity is you can reduce precipitation, and that's a little bit trickier to couple that to sea ice, but the Aleutian Low is this massive low pressure system that sets up in the North Pacific, and it controls winter weather for pretty much the whole Pacific region. And so in years when it's strong, it's going to send storms out shooting across the Bering Sea, heading up to Nome. And when there's a lot of those storms, the sea ice gets pushed farther north, and it's maybe a colder year. The sea ice gets pushed farther north. It's a warmer year. There's reduced sea ice, and there's more precipitation because you're getting more storms. So if you weaken the Aleutian Low, you might have fewer storms in a year, or you might have storms that have less precipitation in them. The other thing you can do is you can actually move the Aleutian Low. And so if the Aleutian Low is farther to the east, then instead of the storms coming through the Bering Sea, the storm tracks are going to come over farther east into Alaska, maybe in the Gulf of Alaska region. And sea ice in the Bering Sea actually expands. And so you get expanded sea ice, and it's more arid up here because there's less precipitation happening. And so the exciting thing about this cruise that we're about to do is that because we're going to be taking these sediment cores, and they're just a time machine going through the sediments, we're going to be able to look at all different times. And so this map is exactly the same map as this one with Alaska and Russia, except now this one is 16,000 years ago. And these 4 green dots are where 4 of the basins that we're going to be aiming to sample from. And so we can look at sea ice in Navarin Basin, in North Aleutian Basin, which has already flooded at 16,000 years ago. And we can compare that to how arid it was in places like Norton Sound and St. Matthew Basin in order to see how that changing sea ice is going to affect the environment and the aridity on land. So I hope you can all tell that we're super excited about this project. We would love to answer any questions that you have. And I'm just going to finish I'm going to leave this up here after I stop talking, because we have a couple of events coming up. So when we're about to get on the ship, we have a couple of in-person events in Nome. One is a community event, and one is a junior ranger event for kids. And then once we're on the ship, we'll have a ship to shore live broadcast that will be on the internet. Anybody can join. It'll be sort of towards the end of our cruise, so we'll probably know what we're doing by then, which will be good. And if you keep your ears out for these, you can follow us on Instagram if you want. We're at Bering Land Bridge. And the Instagram is not really happening right now, but by July, we will start posting. Thanks.
All right. Yeah, I'm excited about your cruise. Holy cow, you guys are a great team. You're hitting it from all different angles, and wonderful. We can't wait to hear the rest of the story. Doug, go ahead. I see your hand is up.
Yeah, that was a great presentation. Really enjoyed that. Really understood it too. Just presented really well. I'm curious whether there's any other locations that have similar studies that have gone on in the past. And I'm kind of thinking maybe Doggerland that joined Britain with, or maybe in Malaysia area, something like that.
So I do know that there have been some pretty cool studies done in Doggerland. I'm not super familiar with them, but I heard a talk once about them. So I know that they're out there. There's also, I've heard about a core like in the Gulf of Alaska region that also transitioned from marine into, or actually, I guess it went the other way because of uplift. But yeah, I mean, there are other studies like this, but I don't think there's anything quite as unique as the Bering land bridge and having this massive continental area to be able to study as sea level rose. I don't know if anybody else wants to chime in on that.
I could just chime in one more thing on that. I know in some of those places, Doggerland in particular, fishermen would drag up mammoth bones and things like that. So is anyone familiar with that kind of thing being dragged up along the shelf there, the Bering Sea shelf?
This is Gay. I've been on a research vessel in eastern Norton Sound, about 6 miles north of Stebbins in southern Norton Sound, southeastern Norton Sound, southern Norton Sound. And we snagged something and had to really back down and get free of it. And when our trawl net came up, we had a pristine mammoth tooth in there.
Pretty good.
Quite a find. Not something you expect when you're doing a crab survey.
Very cool.
Before we get to you, Rick, I want to say, because I forgot to say it, that this is the time for the audience. While you're thinking of your questions, it's always good to give Beth, David, and Sarah a little straight science love. It is never easy to be a straight science, any kind of public speaker. And on top of that, we've got Dave at, what is it now? It's got to be getting on midnight to you, 11:25. And we appreciate you guys all over in whatever time zone you're in giving us this information. So I'll be sure to give them a little kudo if you liked their presentation. It's always appreciated.
You're so welcome.
All right. Rick Toman's got his hand up. Go for it, Rick.
Yeah, thanks, everyone. That was really, really informative, interesting, learned a lot. I did have one question. I think Sarah had showed a graphic showing showing some existing data from land of the type of environment. And there was some dates attached to those dots as well. And what I'm interested in, so you're gonna extract these cores here on your work this summer. How narrow will you be able to date them? And my question is really, presumably during the time that where you're getting cores were water bodies surrounded by land, bogs or lakes or whatever. Undoubtedly, the climate wasn't static that whole time, and so if the window of the dating is over thousands of years, how do you construct a synchronous picture of the environment? Thanks.
We can do the dates. We're going to be using mostly carbon-14 dates dated with an accelerator mass spectrometer, which can be a little more precise and give you a narrower margin of error. What we hope to date, especially when we're looking at sediment that was in lakes, perhaps on the Bering land bridge, we look for terrestrial plant macrofossils. This avoids them taking in any old carbon from maybe the lake itself during their lifetimes. And we should be able to get dates plus or minus less than 100 years. So our dates should be, and often we can get dates depending on the size of the fossil material, or even if you have some charcoal, from a fire that piled up in the lake. You can separate out some of that and date it, and dates can be plus or minus 20 or 30 years. We can also, if we're lacking those kinds of plant fossils, date the pollen grains themselves. If we have high enough concentrations of pollen grains, those can also be dated, and they can date fairly small samples. And again, plus or minus less than 100 years. So we won't be having such a wide margin of error that we can't say that sites, we're not going to be saying sites are the same age within a few thousand years. They're going to be the same age within a few decades.
Okay, thanks for that clarification. I guess I didn't, I figured that with current dating, the individual sites would be tight. So if I could just clarify my question. So
Yes.
you'll have one site that's maybe 14,500 years old,
Yes.
and another site that's 17,050 years old, kind of thing. Presumably the climate may have been different at those two epochs.
Well, we will have cores, so we'll have younger stuff on top of some of those older dates. And we will be setting, obviously there's probably going to be a sort of a gap or a hiatus during the time that that site was flooding. You probably, when the ocean flooded that area, there's probably going to be some erosion and removal of sediment that was previously deposited. But other than that, there should be a fairly continuous record in the marine part of the section. There should be a fairly continuous record in any lake. And we will be setting up a chronology sort of between dated horizons to try to figure out, that we don't have to date absolutely every layer to figure out how old it is.
Very cool. Thank you.
A few dates, and we set up a chronology, and that way we can compare samples from anywhere in our chronology or find something that's the same age in another core, even though that's not the exact date on a horizon. The other thing we will be doing in order to add some additional dates to these cores, we are in discussions with the Alaska Volcano Observatory to, if we find any volcanic ash, which we certainly expect that we will out there, ash that blew over the Bering Sea and was deposited, there is a way to geochemically fingerprint that ash. You can't really date the ash directly. Often it's somewhat weathered if it's been buried in a marine site, but they can have a technique for geochemically fingerprinting it. So if it matches an ash deposit from a known eruption of a known age somewhere, then we know how old that ash layer is.
Very cool. Thank you very much.
All right. I don't see anything in the chat yet. I had a question. Oh, I'm going to go quick, Dean. I had a question for Sarah, and that is thinking about you said it was unexpected that stop me if I'm wrong, you found the kinds of pollen that would indicate it was arid close to the edge of the ocean. Is that right? And that was kind of confusing.
Even a fairly recent study on St. Paul Island, that's when they were, dating some of the youngest mammoth populations that lived there. They also looked at the pollen, and it looked relatively arid and like there weren't a lot of woody plants living there, if any.
So, okay. So my question is, not when you got Rick Toman, he's talking like the climate, you guys were just talking about thousands of years. Would it have been different so that you might have gotten like strong northerly winds that would have blown the pollen down, or is that something you can figure out with your?
It's tricky. That's one of the reasons that more recent study on St. Paul Island used DNA, plant DNA in conjunction with the pollen, because they had very small amounts of things like spruce trees and some other woody plants, but they didn't find their DNA. So that would suggest that maybe it blew in from, and it's hard to say how far away, but that it wasn't really local. But it is possible, if Dale Guthrie's right about low-lying cloud cover in maritime areas, that the vegetation might be different even 300 meters up a hill. If you're above some of the low-lying coastal fog, for example, you might have a more arid site than you do lower down. So that's another reason we want to, looking at an island in the Bering Sea today, that was a high spot. It's possible that the lower elevation areas on the coast had different vegetation, and in fact, it's probably likely. but we don't know what it was.
All right, thank you so much. Dean Stockwell, you've got your hand up. Go for it.
Yeah, two questions, one to Sarah and one to Beth. So Sarah, you said you're going to be doing DNA sequencing. Are you doing both 18S and 16S sequencing?
I am not doing any DNA sequencing. We are working with Beth Shapiro at UC Santa Cruz. She will be doing some of that DNA. So I can't answer that question. That is not my field.
But some analysis will be done. And my question with that regards to harmful algal blooms. And if you can have that person look specifically at Alexandrium types of blooms, I'd really like to see that data kind of processed in your cores. So the other question to Beth, you're
Wait,
doing all
Dean,
the
can I
work.
just ask you a question before you finish that? So Alexandrium, which is a dinoflagellate, is that?
Yes, that's correct.
Okay,
So
go ahead.
that's a nasty one up in the Bering Sea. Down in the southeast, we've had it since the 1600s. I'd love to see somebody come up with data saying it's always flowed through the straits, and your data may be able to show that. So my question to you, I am a diatom person at heart here at UAF. And so when you're looking at the ice algae, I would love to see that information processed. I mean, it's very important to me because I think that ice algae bloom that we have in the spring is very important to the benthos. And since I'd say 2015, that's been kind of changing. And I'd love to see what it was like back in the day. So, I wish you luck in your processing, and I'd love to see that data when it gets processed.
Cool. So there's one thing I just want to say to that, or maybe two things. I don't know how comparable the paleo is to the modern data because you have this time scale issue, which, Rick sort of brought up a little bit, but Sarah's, although our ages are really accurate, marine sediments are really mixed because the benthos is down there mixing stuff up. So, I sort of think about those sediments as being like 100 years or so, like mixed within a centimeter when we're looking at them. So they're not, whereas if you're looking at like things changing since 2015, like that's a really short time scale. So we're not going to be able to like pinpoint things that closely. There's also a little bit of a mismatch between what lands in the sediment and what you see in the surface ocean. But I do also want to say that I've been in contact with Seth Danielson and his sediment traps. And, I have a PhD student who actually looked at one of those sediment traps. And so hopefully she will publish that data. And so the species that I showed with the in the different seasons are actually from her dissertation. So there's a little bit of data out there that maybe I can share with you soon.
When that happens, can you email me that information? I'm here at UAF.
Yeah, totally. Absolutely.
Yeah, I think that's fantastic stuff. And I think some of that sequencing can help you a lot to kind of pull things together. So I think you guys have a great project. Really appreciate the information you're presenting tonight. So thanks.
Cool. Thank you.
And Dean, while you're at it, if you could drop your contact information in the chat, either publicly or privately, that would be probably easy for Beth to add it to her.
You got it.
Stuff. All right. Thanks, Dean. And let's see, Rick. Rick, your hand is up. Go for it.
So from the climate and meteorological perspective, kind of the higher latitude perspective, the idea that somehow there's a persistent marine layer and at higher elevation it would be a drier climate is extremely suspect to me. We have a contemporary example, right? when you're in Nome, take a drive up to top of Anvil Mountain. You've got a nice view of the sea there, and you're more than 300 meters above sea level. And the vegetation is different, but not in that direction, right? Because of the wind pressure up there, and it's cooler in the summer. So I am extremely suspect of that. The meteorology at high latitude is completely different, than the marine stratus of California. So take that for what it's worth, which is probably nothing. But again, we have a contemporary example right at Nome.
No, Rick, I think that's a really good point. And one of the nice things about this study is it's the first time we're actually going to be able to test that hypothesis. Whichever way it turns out, I expect the vegetation will be different, but we really don't know what the lowlands of the land bridge were like. I appreciate your input. That's fair enough, but I'm not invested in it being any particular way. I just want to know what it was.
All right, so there is a question in the chat, and it says it's to Beth from Sally Zellers. While you're focusing on marine sea ice, what type of insights can the team get from looking at seasonal lake ice?
So that's an interesting question that I haven't really considered, Sally. But certainly it could be something to look at. I mean, I'm not a freshwater diatom person at all, so I don't even know whether there are specific diatoms that associate with lake ice versus open water. Probably. I mean, why wouldn't there be? So yeah, I mean, and I think, well, there's been some work about the lake ice, like at Lake Elgagikin, and looking at what that lake does with it has, like, if it has water around the edge or if the ice completely disappears and stuff like that. And so maybe we can use some of their lessons learned to apply to our lakes.
Okay, thanks. I put my thing in the chat because I've never figured out how to raise my hand.
I'm with you, Sally. Oh, and Megan Gannon has her hand up, so she's much more hip to all the Zoom.
No, I'm
Megan
apparently not. But thanks so much for this presentation. That was really fascinating and great. I guess this question would be for Beth. This is just something that was on my mind since I had just written about it for the Nugget. I wrote about this new study that came out a few weeks ago about microplastics being found in sea ice associated algae. So I was just curious if that is something you might be anticipating finding in some of these benthic sediments, and I don't know if you're at all, just thinking about what you might take from that.
Yeah, I mean, I would definitely keep an eye out. I feel like that would only be in the very, very top sediments. But certainly if we found any, we would be able to see them. We're going to make slides and look under the microscope. And so if there's plastic in there, we would definitely be able to see it. And I think that if we did see something sort of, universal across those top sediments, we would definitely be interested in looking at that more.
All right. Thank you. And for the team, I would say that Dean had asked the question about the Alexandrium, and know that there's a lot of interest right now in that, because last year, I don't know if you're aware, but in this region, we had they're telling us globally, it was the second highest level of dense, massive, persistent, and most toxic, harmful algal bloom from the northern Bering Sea to the southern Chukchi Sea from July to September. It was sort of chugging along through all that. And the questions are coming in now, is this something that's coming back, or is this totally novel to the region? And there's a lot of thoughts and work that needs to be done. So know that it might be something you might be able to see. If we've had these the cell counts were amazing, like over 150 to 160,000 cells per liter, that kind of thing. So I don't know if that lasts, but if it's something you can look at, I think there'd be a lot of interest anyway, locally.
So we will have some monitoring equipment on the ship that will be monitored by a research group at Woods Hole and a research group at NOAA. And
Yes.
they're looking for that evidence of another harmful algal bloom. And so they'll be sort of monitoring that in real time while we're out there driving around doing our thing. So, for sure that will be kept track of. Unfortunately, I don't know if Alexandrium, I'm going to actually take a look at the literature, and I don't know if Alexandrium leaves anything behind, any kind of hard part that we can find, but it'd be interesting if they did. That would definitely be something to look for too.
All right. Yeah, thank you. And we're all appreciative to the SIKULIAQ Marine Program and Woods Hole for getting that, and for all of the PIs involved this summer for keeping that on and giving us real-time cell data, Alexandrium or algae data as it goes by. There is a question in the chat box from Sally. She says it would be interesting to know whether algal blooms limited animal migrations in the past during the LGM. I'm not sure what the LGM stands for, but yeah, harmful algal blooms. Last glacier maximum. Thank you. I'm not hip to this. That's very good. Thank you.
Well, mostly during the last glacial maximum, if all goes well, we're going to be sampling primarily freshwater terrestrial sites and not marine sites at that time. But as the flooding was going on, that's when maybe we'll be able to pick up some information about algal blooms by looking at the marine part of the section.
And there's also farther south in the Bering Sea and Sally, in the Gulf of Alaska, and then, pretty much everywhere coastal Pacific, there are these huge anoxic events where the oxygen sort of disappears in much of the Pacific Ocean at certain depths. And so, that happens much deeper than where we are. But I sort of think about that when I think about, sort of algae limiting animals moving around I don't know. Maybe I'm not making any sense.
No, you are. And that makes sense. I was just thinking about the terrestrial organisms and, while you're flooding, and if they're finding things if that was a barrier, in other words. So that, I guess I had another question since I'm talking. And I don't know whether it's for Sarah, but is there a way to distinguish a natural forest fire from a anthropogenic forest fire? In other words,
not that I know
a
of.
hearth or
The way
something.
that we can see forest fires is through the charcoal, but that just means plants burned.
Okay.
So we wouldn't know what started that fire.
Okay.
One thing that we could do, which we are not currently planning to do until we get some more input from the community. We really want to talk about this during our event at St. Joe's. One thing we can look for, in addition to other animals, mammoths, bison, reconstructing the ecosystem, we could also look for human DNA around some of these lake sites. Again, we don't want to do that until we've gotten some feedback from communities close to the Bering Sea, close to the land bridge. We'd like people to be able to ask questions about that, but we wouldn't be looking for remains of humans. We're not going to get that. We're not going to find artifacts. What we would be looking for was whether humans had defecated in the landscape, and that's where the DNA would likely come from. But if fires picked up when we start picking up human DNA, that might be one way to potentially link the two. But other than that, I can't think of a way. And again, until we get some community feedback, we are not planning to look for human DNA.
Okay, thanks.
And you made me think of a question. So we have a tremendous, the Bering Strait region, right, has been occupied for a very long time. I think you guys are the ones that probably get that the most. And we do have tremendous knowledge in the oral history. We have stories that sort of start off with when the ocean was lower or when the ocean was higher. Are you guys at all incorporating as kind of a dive into the literature to look at some of your this, what was it like kind of a thing for either in the literature or probably in the living history that is among us here in the Bering Strait region. It's magnificent.
I
That's something
have
you're,
thought of that, and I know there are some oral histories that talk about when the water was rising and things like that, and I'm extremely interested. I would be especially interested in current oral histories. We don't have a plan for how to talk to people about that at this point, but that's certainly something that we could do, and I'm very interested in those oral histories. Beth or Dave, do you have anything to add?
With respect to oral histories?
yes, long, oral histories that are talking about, are you guys going to do a dive into looking at interviewing people or doing, going back in the records, however you do that, for some of the oral history in this region where you have stories that are about when the water was lower or higher? I mean, really long.
Yeah, no, it had not occurred to me, but I can say that in the Washington, Oregon coast, the oral history there has turned out to be quite correct in some of the very large tsunami that hit it's 1700, and that was well into the history of those people, but it wasn't really understood until other data came along that, by gosh, they saw this and it got recorded in their stories and in their memories. So it wouldn't surprise me at all.
Yeah.
I think this is another thing that would be interesting to talk about during our community event prior to the cruise, and see what some folks in Nome might volunteer and what they know about these oral histories. I think, if we strike up a collaboration, this could be a great graduate student project in collaboration with, our microfossil research and isotope research and DNA research.
Yeah, I would be super, super interested in finding any contacts to have these conversations with, for sure.
All right. So let's just, one last question. If anyone else has any question, now is the time to speak up. All right. Well, my question is, you said there was a barrier between keeping the animals back. And when you showed the list of the animals that sort of stayed on one side on the US side and others didn't cross, and I can picture what a woolly rhino must be. I can sort of picture a short-faced bear and certainly a muskox and things like those were familiar. But what is an American, I don't even know how to pronounce it, KIANG?
I thought you might be asking about the camel for a minute, but no.
No, I can picture what a camel looks like, but I mean, I'm sure it's different than what we got, but kind of, and the horse and all that, but.
I'm about to answer, and I better double check my response on Google because I have looked this up before.
Okay.
And I don't.
Sorry, no, I didn't mean to get, but it's on the slide, and I was like, I thought it
Yeah.
might be like a word like that. The short-faced bear thing had gone like, oh, it's an American Kyang short-faced bear, but I think it was
Yes.
actually something different.
I saw it too. Yeah.
Yeah, I it's a hoofed thing.
Okay, I can look it up. I just wondered if you knew, because I was like,
I've seen pictures of them, but I don't want to call it like an antelope-like thing because that might not be.
okay, it's
correct.
got legs. It's got legs and some sort of horn.
Yes.
Hooves. Yeah. Okay. It's got legs. All right. Very nice. You're still getting a lot of kudos. If, as I guess we'll bring this to a close unless there's any other questions. Thank you for getting over the mark, and poor David needs to go to bed because it's got to be, he's almost at the stroke of midnight. So thank you.
I'll just get some coffee and declare it the new day,
There you go. There you go. Well done. We want to thank everybody who participated tonight. And next week we have on the 31st. Oh my gosh, I'm gonna have to look at, because yes, we have fin whale research. That is going to be by Erica Escajeda, the University of Washington. She's looking at subarctic baleen whales. We're seeing a lot more species now than we have in the past that are coming up. What does that mean? And she's also trying to get at some of the more ancient history. Are the whales recovering and coming up, or is it climate related, that kind of thing? So that'll be next Wednesday on the 31st at 6:30. So We hope to have you join us for that because she's going to be talking about in the Bering Strait region. There's a lot of change going on there with whale abundance and distribution and species. And David, you should go to bed, but unless you want to stay, I'd ask you, all three of you, to stay, and we'll let everyone else take off.
Okay,
we'll
and
see
Jay,
you
I can answer your question. I looked up the Kaeng and
Oh,
I've
okay.
done this before, but it's related. It's a type of horse, basically.
Okay, all right, so like a hooved
It's
horse.
a type
All right.
of what? What?
Horse.
Horse.
Related to the horse.
I wasn't gonna guess that one.
Yeah. All right, well, thank you all so much for coming tonight to Straight Science, and we'll see you next week. Have a good night, everybody.